Hi, welcome to this webinar. My name is Mara Couto-Rodriguez. I am the Director of Research and Development at Biosha. And today I will be talking about hybrid capture SARS-CoV-2, NGS assay, and COVID-Dx, a robust end-to-end solution for viral detection and surveillance of genetic variants. So first of all, I would like to start by introducing myself. So I am originally from Puerto Rico, and that's where I started my research career. I started working with plant microbe interactions, particularly the microbial communities that were associated with seagrasses, and we were employing in this time early metagenomic techniques and classic microbiology techniques to try to identify the microbial communities associated with the seagrasses. And so in the left-hand side, you can see a picture of as electron microscopy of a cross-section of a seagrass leaf, which is showing also some bacteria growing among it. And so after I finished this time in Puerto Rico, then I continue graduate studies at the University of Georgia. While in Puerto Rico, I was introduced to fungi, and I became really fascinated by these types of organisms just because they are unique in their biology and their genetics. And so I moved on to working with the filamentous fungus Aspergillus fumigatus. This fungus is an opportunistic fungus, and we were interested in looking at how some cytoskeletal elements are important during cell cycle regulation, in particular how they are involved in regulating polar growth, which is really important when fungi are invading human tissues. So here in this work, I employed multiple techniques such as classic genetics, cell biology, and then also pyrosequencing. And in the middle, you can observe just a fluorescent microscopy picture of looking at the fungus and how it develops throughout all of the developmental stages. In green, we have labeled septin, which is a cytoskeletal element really important for this transition from polar growth in filamentous fungi. And then in red are the nuclei that are labeled. So after I finished with this research, then I moved on to Columbia University, where I was working with multiple projects in the microbiome field. In these research, I was employing next generation sequencing techniques in order to try and the role of the microbiome during health and disease, both in chronic and acute illnesses, including HIV, stillbirth, bacterial vaginosis, colorectal cancer, and maternal antibiotic exposure. This research was not only limited to the human condition, we also published a paper where we studied the gut microbiome of gorillas from the Republic of Congo, and we were able to identify how the microbial communities in the gorillas shifted depending on the diet that the gorillas were consuming during their seasonal fluctuations. So our most recent paper we have published was also looking at post-infectious hydrocephalus in Ugandan children, and we were able to identify this bacterial strain, Paenibacillus, which was one of the mediators of this hydrocephalus for Ugandan children. So with that, in 2019, I joined Biosha in order to use the skill set that I have acquired throughout all of these years and complement Biosha's mission, which is fighting infectious disease with genomics and AI while building the world's leading microbial sequence database. So it's no surprise that when the pandemic hit last year, we decided to shift our focus and try to help use our skills and the tool sets that we have developed as a company to try to help the pandemic response. So in this talk, you will be hearing a bit more about BIOSH's effort for this pandemic. And so just to line up our agenda for today, I'm going to be talking about how next generation sequencing is crucial for tracking viral evolution to support pandemic surveillance. I am also going to be going into detail on how a hybrid capture NGS workflow looks like. And in addition, I'm going to talk a little bit more about how a hybrid capture-based NGS approach enables more robust and reliable variant detection. So we are a year now into the pandemic, and recently we have started to hear about these new variants that are emerging. And during this time, back in December, we started hearing about B117, a variant that was detected in the UK. But shortly after that, we also, scientists started to report new variants emerging in South Africa, such as variant B1351, variants in Brazil, such as P1, and then also it's no surprise that we have also detected variants in the US because there has been a rampant infection rate during last year. So we've detected variants in New York City, California, and so on. But we do believe that there may be other variants that are circulating among the US population that are still going undetected. So why are these variants concerning? These variants are concerning because they impact our tools that we have to detect the virus, to also eradicate the virus based on vaccines, and it may also impact clinical outcomes. For testing, we have observed that some of the qPCR tests that are used for diagnostics have experienced false negatives due to the mutations that have been arising in some of these strains. Additionally, it is no surprise that as the virus keeps mutating and accumulating mutations, some of these mutations are going to fall within the antigenic region that is used for vaccine development, and that can definitely impact the efficacy of the vaccine. This has already been observed as a 6-fold reduction in neutralizing titers for B1351 for the Moderna vaccine. And in terms of clinical outcomes, not only the virus can mutate and become more transmissible, but also it can become more virulent and then impacting potential the clinical outcomes of patients. So what are some of the key testing approaches that we have available at the moment for SARS-CoV-2? The most widely used are PCR tests, and these PCR tests basically take the nucleic acid, and then we amplify 1 to 3 targets in the viral genome. This is only detects presence absence of the virus within a sample. And so this is a highly scalable and rapid test to use for high volume of samples. However, there is also sequencing-based approaches that that we are able to sequence the full viral genome and allows us to detect changes in the nucleotides, allows us to detect new variants that may be emerging, and also track the evolution of the virus while also comparing viral strains among different populations across the world. Of the sequencing-based approaches that are available at the moment, there are 2 types. There is shotgun sequencing and target enrichment sequencing. If we think about a clinical specimen, in particular a clinical specimen that is used for SARS-CoV-2, is a nasopharyngeal swab. In a nasopharyngeal swab, this is a complex matrix where you're having not only the virus of interest that you want to detect, but also you have human cells and microbial cells within the sample. When you use a shotgun sequencing, you basically extract the nucleic acid of that mix of cells, and then you prepare the samples and sequence everything that is present within the sample. This means that if you think about a virus is very small, has a very small genome, and then it is kind of hard to find the virus in between the high background that there is of human cells, microbial cells, and all of that. So basically, you're trying to find a needle in a haystack. This will require higher sequencing depth. It has decreased sensitivity due to the presence of human nucleic acids and microbial nucleic acid. Another, if you think about then targeted enrichment approach, basically what you're doing is trying to take out first all of those viral sequences by amplifying the target using probes or primers that would target SARS-CoV-2. In that case, you're amplifying that signal, then it would require less step of sequencing, it would provide greater coverage, and would allow for variant detection. This is also less costly and much more sensitive than a shotgun sequencing approach. Of the targeted sequencing approaches, there are two types. There is amplicon-based, and the most widely used at the moment is ARTIC. And in this type of approach, basically there is pools of primers that target the SARS-CoV-2 genome and tile across the viral genome. And then after amplifying the signal, then the sample is sequenced. The hybridization capture-based approach uses a different technology, and in this case, it uses probes that are biotin labeled, and these probes kind of pull out or fish out the viral sequences, and then the signal is amplified, and this has been proven to be more suited for detecting variants, but is definitely a slower workflow. So, as I said, BioSha, we have shifted our attention to helping combat the pandemic. And with that, we partner with Twist Biosciences, and an agile team. We responded to the pandemic, and within 4 months, we were able to optimize the Twist SARS-CoV-2 assay. We validated it on clinical nasopharyngeal samples, built a cloud-based software, and also co-submitted this technology to the FDA. We were able to launch our platform as a research use version back in November 2020. And at the moment, this assay is being used across Europe and US labs to help track and surveil genetic variants. Most recently, we finally received EUA approval for the first hybrid capture-based NGS FDA EUA test. And this has both a diagnostic and research application. Within our solution, we're able to provide deep insight into genetic variants. And it's also coupled with a user portal that is connected to a LIMS system that automatically reports to New York State. So how does this workflow look like? So we have provided an end-to-end solution from receiving of a specimen down to translating it into a clinical report. We start, as I said, we have validated our assay in nasopharyngeal specimens. So we receive nasopharyngeal specimens in transport media. Those nasopharyngeal specimens undergo nucleic acid extraction using the Zymo-Direxol DNA and RNA mini prep kit. After that, RNA is converted to cDNA using NEB enzymes, followed by library preparation of TruSiC compatible libraries using the Twist library preparation kit. Following the library preparation, then the target enrichment occurs using Twist SARS-CoV-2 probe panel. And finally, The pools are loaded into a NextSeq 550 from Illumina. After the sequencer finishes, then these FASTQ files are uploaded through the user portal, BIOSH's user portal. And then this user portal would take the genetic information, it will compare it to a database that we have built, and then we'll generate a report that is very user-friendly. And in this report, we're able to report variants that are detected. We give a viral clade estimation and phylogenetic tree with a background of over 3,000 sequences that we pulled from GSAD. If we look in more carefully into what the wet lab component of this workflow looks like, it contains 5 modules. The first module begins with the nucleic acid extraction. After extraction of the nucleic acid, then it is converted to cDNA, first strand and second strand synthesis. After cDNA generation, then we move on to library preparation. Our library preparation undergoes enzymatic fragmentation, followed by adapter ligation, and then barcoding of the libraries. This will allow us to run 96 samples at a time. And then after the cDNA library generation, then the samples go through the capture, in which we are enriching for SARS-CoV-2 sequences. These samples are pooled in 8 plexes, and from 8 plexes, then there is hybridization to the probes, followed by a post-captured PCR amplification, and finally, libraries are loaded into the NextSeq 550. So we, as I said, we have validated our results on nasopharyngeal specimens, and now I'm just going to talk a little bit about the performance of our assay and also other results that are associated to our assay. So first of all, we compared our assay to an RT-PCR, and our assay yielded 96.7% positive agreement and 100% negative agreement to the RT-PCR. In addition, we established a limit of detection of 800 copies per ml, which is 0.8 copies per microliter. This is a little bit more sensitive than other sequencing-based assays that are in the market at the moment. Within our report, as I mentioned, we are able to also covariance on samples, and this is in the bottom is an example of one of the figures that is generated in our report. You can observe the structure of the SARS-CoV-2 genome. In the top panel, we report the coverage of the sample, including the variants that were detected within the sample. In this case, the sample contained 6 variants, and in the bottom panel of it, then it's this comparison against the database that we have built, and then it also shows the frequencies of these variants detected within the sample when compared to the global database. Additionally, we wanted to understand the performance of our assay compared to the gold standard, which is RT-PCR. And we used, we took our data and broke it down in CT values categories from 15 to 20, 20 to 25, 25 to 30, and 30 to 35. And we were able to detect an average fold enrichment of the virus of 50,000x for all the samples within our clinical validation. We detected a 93% coverage at 5x across the genome, with 99% coverage in 48 out of the 60 samples within the clinical validation. When we looked at the depth of coverage, our percent of coverage of the genome at different depths, we are able to see that any CT value below 30 gives really good enrichment of the viral sequences and allows us to have very good coverage to confidently call variants of all samples. Even looking at a depth of 100x, we're still getting at the higher range of 25 to 30, a very good coverage, percent coverage of the genome. So we additionally took these samples along with a small subset of samples from Tennessee, and these samples were analyzed against the global database for SARS-CoV-2, and we were able to detect a total of 730 mutations within 71 samples that we tested, and these mutations were located at 258 different mutation sites. Of these, 107 mutations were new mutations that were not previously reported in GSAT, and 24 of these mutations were located in the clinically relevant spike protein. The most frequently detected variants within the sample set were C241T, which is an extragenic mutation, and then D614G mutation in the spike protein. This is one of the most prevalent mutations among SARS-CoV-2 samples since late spring. So we additionally, as I said, we use samples from two different geographic locations, New York City and Tennessee, and these share similar genetic mutations. And in the picture and on the right, you can see on the top panel the SARS-CoV-2 genome and all of the 71 samples that we have analyzed. These were aligned against the Wuhan reference strain. And we looked into what were the mutations that were present among all samples. In purple, it is denoted all of the new mutations that we found within our clinical validation study. On the top panel, you can also see the percent of samples that contain those mutations. So basically, we had some handful of predominant mutations. Of those, as I mentioned, we had D614G was detected within our cohort and was one of the most prevalent mutations within our samples. Additionally, the majority of the novel variants that are denoted in purple are located at the ORF1ab gene, and 24 of those novel variants were detected in the spike protein. We also detected 14 frame shifts after synonymous mutations, and 2 of those frame shifts were detected in the spike protein. We identified unique mutations specific for the geographic location, particularly for Tennessee samples. We were able to detect a mutation in the ORF1ab region, which is mutation T6394C. And we also took some of these new mutations that we detected and were able to validate them with another sequencing-based approach, which was Sanger sequencing. This confirmed that our technology is quite robust at detecting variants, and then also it allows us to confidently call all of these new mutations that have been found. So additionally, within our assay, we are able to study the microbiome. And this is because the extraction kits that were used on our validation, and that is now established in our workflow, allows for DNA, separate DNA extraction. And so with this in mind, and because we work biome at BioSha, we were very interested in looking and profiling how the nasopharyngeal microbiome looks like in patients that have that are positive for SARS-CoV-2 and patients that are negative for SARS-CoV-2. So we were able to profile the microbiome in 48 nasopharyngeal swabs. We had to exclude some samples within our cohort because most of these samples yielded very high human background and there was no detectable microbiome. However, in this figure right here, you can observe a heat map that is denoting the relative abundance of the top 50 most abundant bacterial species. These bacterial species were not, the microbiome profiles were not separating depending on COVID status. However, we were able to use other metagenomic tools to determine biomarkers within our samples, and we found an overrepresentation of 6 different taxa, including Prevotella, within COVID positive samples. With this metagenomic approach, we are also able to profile AMR genes and viruses that are present in the specimens. We are particularly interested in AMR genes, given that many patients are being overprescribed antibiotics during this time of COVID, and we need to be very mindful in monitoring AMR resistance genes that may be emerging due to the overprescription of antibiotics to COVID patients. Many of these patients undergo co-infections, and so it is quite crucial to also address the AMR profiles within COVID patients. We detected an antimicrobial resistance gene called MEL-C. This one was overrepresented in COVID positive specimens, and also we were able to detect some DNA viruses, including influenza A and herpes virus 4. So in summary, our approach, we're able to also profile the nasopharyngeal microbiome and gain some insights on whether there are some of these microbial communities that may be modulating the immune system, or also may be important for co-infections on SARS-CoV-2 patients. So it is quite a great tool for any researcher that would want to look into the microbiome. So with that, then I would like to summarize the importance of hybrid capture for viral surveillance. So our hybrid capture approach has the ability to tolerate more mismatches, about 10 to 20%, due to the usage of 120 base pairs double-stranded DNA probes. This allows for a more efficient capture of the target. It enables more robust variant detection as the virus continues to evolve. These mismatches allows us to keep discovering these new variants, which is a limitation of some of the amplicon-based sequencing approaches that are available right now. These use primers of 20 to 30 base pairs in length, which also could hinder any mismatches, could hinder the ability of the primer to bind, and then we would experience dropouts and missing some of these variants. An example of this is also observed in RT-qPCR tests. Some of the RT-qPCR tests that are available in the market targeted the spike protein. And these region has experienced a significant amount of mutations, in particular for the B117 strain detected originally in the UK. And this has yielded into increased false negative results for some of these tests. So what is the next steps for our SARS-CoV-2 assay? We want to expand to different sample types. We have already tested some other sample types, including saliva, in collaboration with other researchers. We are interested also in expanding into a more comprehensive respiratory panel that perhaps would allow us to identify influenza and SARS-CoV-2. And we also want to have efforts to support automation to increase throughput of samples for either research labs or clinical labs. So, and what's the future for BioSha? So Biosha, we are interested in looking forward in enabling personalized, data-driven pathogen discovery and diagnostics from the individual to the community, building precision infectious disease diagnostics and NGS-guided patient treatment that would lead us to build leading database for predictive analytics to protect communities. And also, ultimately, we want to be able to detect any pathogen from any sample type or any type of domain, allowing us to move forward on eradicating death due to infectious diseases. And so with that, I would like to close just acknowledging our amazing team at Biosha. We have all worked very hard on the past year to develop this solution and bring it to researchers and clinical diagnostic labs. And I would also like to thank our collaborators at Twist, which we developed the test with, in particular, Kristen Butcher, Sijuan Shen, Brian Hoagland, Shaquille, and Peter. Our collaborators in the University of Tennessee, Dr. Colleen Johnson and Mariah Taylor, Robert Burstein at Lenco Laboratory for providing clinical samples both for validation and any other follow-up work, Agnes Berkey, she's a consultant to Biosha, and then also IAVI, SUNY Downstate, and Biobad. So with that, I would like to then say thank you for watching this webinar. And if you have any questions or would like to know more about our assay, please feel free to reach out to my email or to any of the social media platforms. Thank you.